A very good day to Dr. Dhoswati. Today I'm going to present about the diversity of marine phototrophs. So let's see the timeline from the traditional to current understanding. At mid-1970, diatoms and flagellates were the major primary producer. And then at late 1970, the scientists revealed cyanobacteria cyanococcus under the microscope. At late 1980, the scientists recognized that the cyanococcus contained chlorophyll A under the high-performance liquid chromatography technique. At 1996, chlorophyll D was found in Cyanobacterium acrichloris marina. At 2000, the scientists revealed two significant findings. One is about the wide distribution of aerobic and exogenic photosynthetic bacteria AAPB, each contain bacterial chlorophyll A, and then second is about the wide distribution of the phototrophs containing bacteria. At 2007, the scientists discovered that the phytoplankton each did not belong to any phylum. At 2010, chlorophyll F was reported and several reports on marine phytoplankton were published. At 2011, rapimones was discovered by the metagenomic analysis. So let's see the detailed explanation what happened at the mid-1970s. Primary production mainly performed by the diatoms and the flagellates. They are classified as the major organisms because they can collected by the phytoplankton net and they can easily observe from the phytoplankton sampling and the method for the phytoplankton detection at the time. Plankton net used for sampling and various types of net mesh such are used, for example 100 micrometer, 70 micrometer, 60 micrometer. Phytoplankton with a cell or colony size of more than 60 micrometer were used in the phytoplankton research. This is the diatom and the flagellate they discovered in the mid-1970s. Okay, let's see about at the late 1970s. At the late 1970s, under the development technique by the microscope and from the electron microscope, they are able to reveal cyanobacteria cyanococcus. This species mainly distributed at the tropical and subtropical region of the marine environment. These species are called as picocyanobacteria also because they add the tiny cells with 0.5 to 2.0 micrometer in length and 0.5 to 1.0 micrometer in diameter. These cyanococcus species act as a significant role as a primary producer in marine environment, but their growth also limited by the concentration of the nutrients and also the trace matrix. Below is the picture of the cyanococcus bacteria. At the late 1980s, chlorophyll A each had an observation spectrum recognized in the cyanococcus. This detection produced by the high performance liquid chromatography HPLS technique for the pigment analysis and of the cell detection technique using the flow cytometry. Other than that, they also found the genus Prochlorococcus. This genus is dominant phytoplankton in tropical and subtropical regions. They are able to perform oxygenic photosynthesis using chlorophyll A or B, which essentially absorbs the blue light dominating. The amount of the chlorophyll A is 3 times higher than of the usual chlorophyll A. Below is the picture of the Prochlorococcus. By the 1996 Cyanobacterium aqueochloris marina, which perform oxygenic photosynthesis using chlorophyll D, was found. Chlorophyll D is a red-shifted chlorophyll which can absorb far-red light for an almost 13 nanometer longer wavelength than the chlorophyll A. The cyanobacteria employs chlorophyll D not only for the light harvesting antenna but also for the chlorophyll in the reaction center. Below is the picture of the cyanobacteria. Okay, a 2000 wide distribution of aerobic and oxygenic photosynthesis bacteria AAPB, which contain bacterial chlorophyll, are found. AAPB derived from the non-sulfur purple bacteria, the anaerobic and oxygenic photosynthesis bacteria ANAPB. ANAPB and AAPB have some differences. A and APB distribute normally in an anaerobic environment, while the AAPB distribute in an aerobic environment. A and APB they have to perform photosynthesis without the oxygen, and AAPB have to perform photosynthesis with the oxygen. These both bacteria have the similarities. Each is they contain bacterial chlorophyll A. The in situ growth rate of the AAPB in seawater faster than the other heterotrophic bacteria. Another significant finding is the wide distribution of the proto-rhodospin containing bacteria. Proto-rhodospin is the rhodospin-like protein detected in the marine proteobacteria. Rhodospin is a biological pigment which acts as a primary photoreceptor in the vision. On-rhodospin-like protein was known to contribute to the ATP synthesis. For example, the bacterial rhodospin of Allobacteria in archaea, which can transform the light energy to transmembrane proton electrochemical gradients for ATP synthesis. Proto-rhodospin was widely and significantly distributed and able to act as a significant role in light energy transformation in the marine environment. At 2007, phytoplankton, which did not belong to any phylum of algae, were discovered by the metagenomic analysis. For example, is the Picobellifer. Picobellifer is a unicellular heterotrophy, each a size of less than about 3 micrometer. These algae distribute widely in the coastal areas of the Europe and North Atlantic. That is the picture of the Picobellifer seen under the microscope. A 2010 chlorophyll F, which is a red-shifted chlorophyll, was reported. They are found from the cyanobacterium were isolated from the microbial mat on stromatolite at the coast of Australia. This absorption occurs around the 720 nanometer and can also far red light up to around 716 nanometer. Other than that, with the technique of the metagenomic and the flow cytometry, several reports on the marine phytoplankton analysis were published. In this report, they stated that the many undescribed species are found and widely distributed. By the pigment composition analysis, they are able to detect the carotenoid 19HF. At 2011 Rapimones, the new phylum was detected by the metagenomic analysis. This phylum is a type of green algae widely distributed in the coastal areas of Europe and the Atlantic and the Pacific Ocean. The temperature of the phylum ranging from the 52 degree to 79 degree. That is the picture of the Rapimones cells, which can be detected by the high electron microscope. These are the references where I find extra information other than the article. In conclusion, from the timeline of the traditional understanding to the current understanding, we able to know the evolution of the small size organism to the big size organism. Other than that, we also able to know the instrument they use and also the evolution of the instrument from the small size instrument to the big size instrument. That's all from me. Thank you.